Hello learners, welcome to Ahmad Coaching and I'm your teacher Dr. Anam. Today we are going to study about lipid digestion and absorption. The digestion of lipid, it starts from the mouth. In the mouth we have this lingual lipase which is going to convert the fat into fatty acid and glycerol. A fat molecule is made up of 3 fatty acids and 1 glycerol molecules combined together. So we have this CH2O, C double bond O, OH, CH2O similarly. This is one fat molecule. In this molecule, this part here, this is glycerol, while this here is one fatty acid, second fatty acid and third fatty acid. So when this fat molecule is going to digest by the lipase enzyme, we will have 3 fatty acid and 1 glycerol molecule. Small amount of fat is digested in the mouth. From the mouth, the food will move to the stomach. In the stomach, we have gastric lipase. Gastric means stomach. Again, the fat is going to be digested into fatty acid and glycerol. In the mouth as well as in the stomach, a small proportion of fat is digested. The rest of the digestion of the fat, it will take place in the small intestine. So from the mouth, the food, it will enter into the small intestine. In the small intestine, we have duodenum, we have jejunum and we have ileum. These are the 3 parts of the small intestine. In the duodenum, there comes some secretions from the pancreas as well as from the liver that helps in the digestion of lipid. What will happen that from the pancreas, pancreatic lipase, it will enter into the duodenum part of the small intestine. The lipase, we know that it is going to digest fat or lipid. But to assist the digestion of the lipase, the liver is going to send bile. The bile is produced in the liver, but it is stored in the gallbladder, and when needed, it is going to be entered into the duodenum. The bile doesn't contain any enzyme, but it contains salts and minerals. The main function of bile is the emulsification of the fat. What is actually emulsification? For example, you have a glass of water, and on the glass of water you have a layer of oil like this. This oil layer is intact, but you can disrupt this layer by adding some liquid soap. As soon as the soap is going to enter here, this layer is going to degrade. This is actually the process of emulsification, to disrupt the continuous layer of lipid. So when the layer of lipid is disrupted, we will have small fat molecules, and when the small fat molecules are there, it will be easy for the lipase enzyme to act on it and convert it into the product. So bile is just going to help in the emulsification process. As soon as the emulsification takes place, the lipase or the pancreatic lipase is going to act on the fat, and this fat is further converted into fatty acid plus glycerol. From here, the rest of the food, it will enter into the second part of the small intestine, which is known as jejunum. In the jejunum, we have this intestinal lipase, and this intestinal lipase will further convert the fat into fatty acid and glycerol. So we have fatty acid and glycerol. In this way, the maximum amount of fat present in the food is digested. Now the end product, fatty acid and glycerols, they are present in the jejunum. Until now, the digestion of lipid is completed. Now the next step is the absorption of lipid. What will happen that this fatty acid and this cholesterol, they are going to be converted into the micelles. What are the micelles? That these molecules, they are going to be surrounded by the water molecules. So each molecule is going to be surrounded by the water, and this is known as micelles. These micelles then enter into the third part of the small intestine, which is known as ileum. In the ileum, we have this folds, which is known as villi, or for singular villus, and to this villus we have the microvilli or microvillus attached. The microvillus or microvilli are the sites which are going to help in the absorption of the lipid. Now these micelles, they are going to enter into the enterocyte, or this internal part of the microvilli. Here we have these triglycerides. The triglycerides, as they will enter, they are going to be converted into the triglycerides. Then this triglyceride, it will pass through this Golgi apparatus for the packing, and then when it will pass through the Golgi apparatus, it will be transported out in the form of chylomicron. Now what is chylomicrons? The chylomicrons, they are actually lipoproteins. It means that the lipid which was coming from here, it is packed with the help of this protein in the form of this chylomicron. This chylomicron is going to take the lipid from one part of the cell to this borderline, and here it is going to fuse with this villus, and then the lipid or the triglyceride, it is going to enter into this villus part of the small intestine. Here we have this blood vessels going, so this triglyceride is going to enter into the blood, and from the blood it will be taken to its destination wherever it is needed. These blood vessels, or a network of blood vessels, is known as lecithin. So in this way, the lipid is going to be digested and then absorbed into the microvilli, then transported in the form of chylomicron, fused with the villus. The chylomicron protein will remain in the microvilli, but only the triglyceride, it will enter into the villus and then the blood, and it will be taken to its destination. So this is how the lipid is digested and absorbed in the small intestine. So that was our lecture about lipid digestion and absorption. If you have any further question related to this topic, you can ask me in the comment section. If you enjoy this video, like it and subscribe to my channel. I will see you in the next video.